In this video, we're going to take a look at the remaining Mendelian exceptions. Whenever you have a slide with a question or task, be sure to pause the video to attempt to answer it yourself and then listen in afterwards. So explain why it would be unrealistic to use simple Mendelian genetics to describe and make predictions about height. So here we have height in humans specifically. Now, if you remember, Mendel did experiments with plants, and he had tall plants and short plants. And so there were two alleles, a big T and little t. But you'll notice here, we don't really have, tall people and short people. Not all short people are the same height. Not all tall people are the same height. There's a lot of variation within height, and it's not just tall or short, and that's it. You can see there's much more than just one tall and one short. So that can't be explained by simple Mendelian genetics like monohybrid crosses, like a big T, little t. It doesn't make sense. So what Mendelian genetics typically assumes is that if you have one characteristic, it's controlled by one gene only. Now, that's called monogenic inheritance, where a character is under the control of one specific gene locus. So in plants, when you're looking at pea color, for example, that's controlled by the pea color gene Y. Okay, and typically traits that are controlled by monogenic inheritance, they're either or traits, are called discontinuous traits, sometimes dichotomous traits. Basically means they're either or. So for example, the peas could be yellow or green, and there's no other variation in between. But not all characters are actually monogenic inheritance. Not all characters are either or traits. Some characters have a lot more variation within. And an example of that is human height, right? So you're not just 5 feet or 6 feet. There's, 5'1", 5'2", 5'3", 5'4", so on and so forth. That's called, those traits are called continuous traits or sometimes quantitative traits. It's when there's a range of traits that exist. And the reason it happens is because in reality, many characteristics are not controlled by one gene, monogenic, but actually by several genes. It's called polygenic inheritance. So characteristics that are controlled by several genes, not just one, so genes from different loci, those are called polygenic inheritance. So height in humans is an example, skin color. So you're not just black or white. There's ranges of skin color, eye color in humans as well. And this is a reality of many characteristics that exist. So it's not just simple monogenic inheritance. So one interesting thing that we can see about these polygenic traits or these continuous traits is that if you were to plot them on a curve for the population, you get what's called a normal distribution or a bell curve, which looks like this over here. And so basically you have a lot of individuals on the extreme, on one extreme, very few individuals at one extreme, and very few individuals at the other extreme, and most people kind of exist kind of in the in-between area. So you can see that for skin color over here. So there's very few individuals that are very, 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 very light, very few individuals that are very, 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 very dark, and then most individuals are kind of like in the in-between area. Here you can see the same idea, a normal distribution or bell curve for height. And so you can see very few people are in the very, 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 very short end. Very few people are in the very, 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 very tall end. Most of them are kind of the in-between area here. Whereas the monogenic characteristics, the discontinuous one, they don't have that bell curve like you would see here. They have more of there's either this or that. So widow's peak or no widow's peak, and that's about it. So long story short, we can't always assume that all characteristics will have, big T and little t only, and they're only controlled by the T gene or the Y gene. Many characteristics are controlled by a variety of genes, and so to predict your phenotype based on the genotypes is very difficult because there are many genes that contribute to your height or to your skin color or other characteristics that we're thinking about here. And that's called polygenic inheritance, which, again, goes against the idea of the fact that characters are coded by one gene. So it's not true. There are many characteristics can be coded by several genes. There are some coded by one gene, but there are many that are coded by many genes, polygenic inheritance. So let's take a look at how this works here. If you want to make some predictions, we would not ask you to make a Punnett square this large, but just to show you how it works. So if you're looking at humans, skin color pigmentation, is actually controlled by 60 different genes, not just like, big A, little a. There's big A, little a, big B, little b, big C, little c, big D, little d, so on and so forth. 60 different genes. So that's polygenic inheritance, and we can see that based on the bell curve. So how does that work, though? How can you predict your phenotype? Let's look at a simplified example. Let's just go with 3 genes controlling skin color. So A, B, and C. Every dominant allele you have for A, B, and C contributes a unit of darkness. So the more dominant alleles you have, the darker you're going to be. So if we look over here, we have big A, big A, big B, big B, big C, big C. 6 units of darkness. So in this simplified example, this is the darkest you can get, 6 units of darkness. Whereas here you have big A, little a, big B, little b, big C, little c. Each of these uppercase ones, each of these dominants give you a unit of darkness, so that's 3 units of darkness. So it's not quite 6 units of darkness, it's not quite 0 units of darkness. So what would the whitest or lightest genotype be? Well, think about it. Pause the video, think about what the lightest would be. How light could it be? And we should go with, if you look at the Punnett square here, you can probably figure it out. If you look over here, no units of darkness at all. And so that's the lightest you could be over here. So all recessive, no dominant, no units of darkness. Which person would be darker? Big A, little a, big B, little b, big C, little c, or big A, big B, little c, little c. So think about that question very quickly, or pause the video, think about it. Which one would be darker? Think about which one has more dominant alleles. And then from there, you might be able to figure it out. So if you thought about it, you'll probably realize that, okay, there'll probably be equal degrees of darkness because this has 3 dominant alleles, and so does this. So likely equal degrees of darkness. However, we're assuming that each gene contributes equally to the skin color. It's possible that maybe other genes contribute in different ways, but for now, we'll assume that they all contribute equally. So this is just an example of polygenic inheritance in skin color, how it works. In reality, keep in mind there's actually 60 genes that control skin color. So this is just a simplified example. This is a video you can watch later on. It connects skin color to evolution. So just watch it and make a few notes on it to understand the idea behind the different skin colors in evolution. All right, so let's take a look at this next question here. Describe the phenotypes of the flowers below. Propose genotypes for both. Make your own letters. So pause the video, try it out. Phenotype-wise, hopefully you said that this is, pink, and then hopefully you said this was blue. Genotype, make your own letters. It's hard to really determine this here, but if you had something along the lines of, let's say you said this was dominant, maybe, then maybe this is big P, little p, or big P, big P. You could have done the other way around. Doesn't really matter. I just want the idea that, phenotypes and genotypes. And then if this was recessive, this might be little p, little p. Okay. Now, here's a weird thing. These actually have the exact same genotypes. These are genetically the same, yet they have different phenotypes. So another example here, let's take a look at this. What factors determine cholesterol levels in your body? So here we can see blood vessels with little cholesterol. Here we can see blood vessels with a lot of cholesterol. This is a cholesterol molecule over here. What determines that? So pause the video, think a bit. If you said genetics, that's true. If you said what you eat, that's true as well. So there's a few factors there that affects your cholesterol levels. So it's interesting to see that, maybe these two individuals are twins and they have the same genes, but this person has more cholesterol, maybe because they eat, their diet's different. It's possible too. So one of the Mendelian assumptions, essentially, where Mendel was lucky not to encounter this too much in his studies, were that genes and characteristics are unaffected by the environment. But in reality, things are affected by the environment. Characteristics are affected by your environment. So Mendel really studied organisms where characters were not significantly affected by their environment. He had a very controlled place, so there was not much of that effect. It's called nature. Nurture is where characters are affected by the environment. So you might have two individuals with the exact same genotype, but their phenotype is different. So here, these are hydrangea flowers. This is the same genotype as this one here. It's just that they're found in different soils. This one here is in a basic soil, and so it's pink as a result, almost like indicators, eh? And this one here is in acidic soil, so it's blue as a result. Here we have a Siamese cat. All the cells on that produce the fur have the same genes. The thing is that the pigment enzyme that adds coloration, so like the black parts here, doesn't work well at high temperatures. So even though there's the pigment enzyme likely being produced by the genes, if you look at the sort of core of the body where the temperatures are higher, it's a bit whiter there because that's how you have high temperatures and the pigment enzyme doesn't work. So again, an example of, cells with the same genes but having different expressions because of their environment. And then in humans, cholesterol levels, yes, they're affected by genes. Your genetics play a role, but also your diet and lifestyle affect that as well in certain diseases that you might have. So this is just the idea that understand, that there is a nature component, a genetic component, but there's a nurture component as well. Those characters are affected by the environment. Even though you have genes that code for a certain thing, you might turn out looking a little bit different because of the environmental influence. Okay, so in the next video, we're going to take a look at the remaining patterns. For this video here, the patterns that we saw, or the exceptions we saw, are that characters can be affected by their environment, and we also saw that genes can be coded, characters can be coded for by more than one gene, and that was called polygenic inheritance.